Hi, I am SB sir, and today we will be learning about pH. What is pH? It's a very beautiful thing. It's a very beautiful idea of chemistry, and today we will learn that what is pH. Now, what does these two letters mean? The two letters P means negative logarithm and the letter H means the hydrogen ion concentration. The letter P means negative log. You take negative log of a hydrogen ion concentration, and that completes our idea of pH. So every ion present in any solution has a concentration expressed in moles per liter. Say I say about a particular thing, say 2 into 10 to the power minus 5 moles per liter. In biological fluids, there is a range of hydrogen ion concentration. So this range falls. We know that our body pH is around 7.4. So this is actually our body pH. Our body is slightly alkaline and it is maintained at a pH of 7.4. Means, why do we say that this pH is so important? Because when the pH, the absolutely neutral pH is around 7. It is absolutely neutral, means this, if a fluid is around 7, is the pH of a fluid is around 7, say this is neutral. So this is neutral means the amount of hydrogen ion concentration and the amount of hydroxyl ion concentration, water ionizes into these two ions. So they are equal. These two ions counteract. They counteract to form water. Now, if all the positive charges that is contributed by the hydrogen ions and all the negative charges that is contributed by the hydroxyl ions, when water ionizes to H plus and AOH minus ions, these two ions are formed. And these two ions counteracts each other to make the whole thing neutral, and we call that the pH of this solution, which is neutral, absolutely neutral, the same number of hydrogen ions and the same number of hydroxyl ions. The number of hydrogen ions equals the number of hydroxyl ions, and we say that this solution is neutral, and the pH of a neutral solution is 7. So what is pH? pH is something when it's a kind of scale. Say this is a scale. If I draw a scale and if I say that the pH here is 7 and these are the values that goes to the negative side of the scale and these are the values which goes to the positive side of the scale. So here, say I say a value of 10 which is around positive side, and I say a value of 4 which is on the negative side of the scale. So these two pH values 4 and 10 reflect hydrogen ion concentration. The hydrogen ion concentration, and it is majorly a factor of 10 which says this factor of 10 to the power minus 5 means it is around 10 to the power minus 5 means it is in the negative part, means minus 5 comes here more than minus 4. Say if it is 7, it is here. If it is 10 to the power minus 10, it is here. So a hydrogen ion concentration which is 10 to the power minus 10 and a hydrogen ion concentration which is, it is obviously in moles per liter, means a hydrogen ion concentration which is in the order of 10 to the power minus 4 is much stronger than which is at a 10 to the power minus 10. Say 1 mole, say if I write it like this, say if I cross out this 1, 2 and I write it here, 1 into 10 to the power minus 4 moles per liter. I want to explain this first. 1 into 10 to the power minus 5 moles per liter. That means 1 mole is divided into 10 to the power minus 5 fractions, means 1 mole is divided by 10 to the power minus 5. 1 2 1 2 3 4 and 5. So this is 1 mole divided by this amount of this factor 10 to the power minus 5. So 1 mole, it's just a fraction of 1 by 10 to the power minus 5 moles of hydrogen in 1 liter. So the amount, when this 10 to the power minus 5 goes to 10 to the power minus 10, so the amount of hydrogen goes down. 10 to the power minus 10 means 10, 1 2 3 4 5 6 7 8 9 10. I've written 10. 1 mole of hydrogen, 1 mole of hydrogen is divided into 10 to the power 10. So it is just a fraction, 1 by 10 to the power 10 moles. It's a very small amount present in 1 liter of a solution. So this is the hydrogen ion concentration, and more negative this number above, this number, this 5 only reflects the pH scale. This 5 part of this number pH reflects this pH scale, and this 5 is, so more this number getting, if the number is getting 10 to the power 10, so it is lesser amount of hydrogen. If it is from a scale of 7, if I write 10 to the power 5, 10 to the power minus 5, so it is going to this part of the scale. So these actually reflect the hydrogen ion concentrations, the hydrogen ion concentration in moles per liter. So 10 to the power minus 5 moles per liter is much larger than 10 to the power minus 10 moles per liter. The concentration of hydrogen is much more here. So when the concentration of hydrogen is much more, if it is more than 10 to the power minus 7, if it is more than 10 to the power minus 7, that is, it is around 10 to the power minus 5 or 10 to the power minus 4 or 10 to the power minus 3. These are the higher hydrogen ion concentrations, and so these solutions are positive solutions, means they will have a positive charge. So if it is 10 to the power minus 10, so it's a much lesser hydrogen ion concentration, and these solutions are negative solutions, means it has far less hydrogen ion concentration relative to hydroxyl ion concentrations. And because why we call it negative solution? Because they have lower hydrogen ion concentration relative to hydroxyl ion concentration, because at pH 7, just now I told you, the hydrogen ions and hydroxyl ions balance each other, the numbers balance each other to make a perfectly neutral solution. So maybe by now I have explained you why is pH very important, because actually if it goes down, the value goes down to from 7, it goes down to 4 to 6 5 6 5 4 3. So the hydrogen ions are increasing in concentration, because a pH of 7 is this 7, the 7 is actually this 10 to the power minus 7, 10 to the power minus 7, because this is a log scale, because while we say pH, it's a log scale. I'll explain that to you now. Let's assume I take a value of a particular acid of say 4.47 into 10 to the power minus 7 moles per liter of hydrogen. This is the hydrogen ion concentration that I have written here. The hydrogen ion concentration is 4.47 into 10 to the power minus 7 moles per liter. Now how do you interpret this in a pH scale? So what is pH? It is just the negative log of this hydrogen ion concentration. So I take negative log of hydrogen ion concentration of this hydrogen ion concentration. So negative log of 4.47 into 10 to the power minus 7. So it comes to negative log of 4.47, which has a value, which has a particular value. minus negative log of 10 to the power minus 7, which also has a particular value. We know the log of 10 value is 1. So log of 4.47 value is around 0.65. You can just see it in your calculator. What is the value of log of 4.47 and 0.65, negative 6.65, and this negative comes here, this negative cancels each other, it becomes positive value. Log of 10 is 1 into this 7 comes here, 7 into 1, log of 10 is 1, log of 10 is 1 minus 7, and this negative goes away, it becomes positive. So 7 into 1 is 7, so 0.65, negative 0.65 plus 7.0. So what is it actually? Is 7 minus 0.65 is 6.35. So just here we see that a little bit increase of hydrogen ion concentration from 1 into 10 to the power minus 7 to 4.47 rise of hydrogen ion concentration, 4.7 factor. This is the factor which has risen, which has made the hydrogen ion concentration has increased by this factor 4.47, and the pH has got lowered 6.35. And this is how we interpret the values of pH. So let's think of something else. So why is this pH so important? Now I'll teach you why this pH is so important. Let us now consider an acid. Acid is written by Ha. This is the anionic portion of the acid. When this acid ionizes, it gives H plus and A minus. That means it is the anion which is I have written by A minus, and this is the proton, the hydrogen ion. Now, if this acid is a weak acid, then this acid will not completely break into these ions. Some amount of acid will always be in the undissociated form. But when this is a strong acid, this acid will completely dissociate into these two ions, and it will liberate all these hydrogen ions, and there is a very high damage that strong acids cause. Why this damage? This liberation of these hydrogen ions, these protons are very small, because you know the atomic number of hydrogen is just 1, so they are very small positive charges, and these positive charges cannot be they are very damaging, means they will always bind to everywhere and make any kind of molecule positive. Means they are like adhesives, they are like paints. Suppose it's these H plus ion goes, I just drop a ink in a glass of water. The glass will be painted because the ink will now go on and it will attach to the sides of the glass, and the glass will now get painted if put throughout the water, but still the glass look bluish. Just it is this type of things that happens when hydrogen ions are there. The proteins, the enzymes, the biological molecules get painted by hydrogen ions. All the anions, all the ions in our body stays as anions. Means why they stay as anions? Because our body pH is around 7.4. At this pH, all the biological proteins, are slightly negative, and these proteins, say this can be enzymes, this can be any proteins. This slight negativity in the proteins is in their different side chains, and these side chains will get will bind to these hydrogen ions, and that there will be a conformational change, there will be a structural change, because you know the side chains of these proteins are linked by hydrogen bonds, van der Waals interactions, and different types of bonds. And if this calamity of hydrogen, this hydrogen being a molecule which will disrupt your structure. See, if I cover you up with a certain very sticky paint, and I tell you to move your arms freely, to walk freely, you will not be able. You will be having a conformational change. It means somehow your fingers will get attached to your hand and you will not be able to write, you will not be able to do any work, because your fingers are getting attached. So hydrogen ions actually produces this kind of effect. The molecules change their structure. The molecules loosens, some molecules opens up, some molecule closes down. So this hydrogen ion gets painted on those molecules, on these proteins, and they will now create calamity inside. So pH is very important for our biological fluids. Our biological fluid, every biological fluid has a particular range of pH. Means there are different biological fluids in our body. Apart from blood, there are lymph, there are aqueous humors, vitreous humors in the eyes, there are CSF in the brain, cerebrospinal fluid, there are urine, you have urine as in your urinary bladder, that is also a biological fluid. Then there are different extracellular fluid, intracellular fluid, all these types of biological fluids. Every organ has a particular type of fluids inside every organ. So say the kidney has a biological fluid. What is the biological fluid? It is a glomerular filtrate. It is somehow different from the blood. So the filtrate is also a biological fluid. So all these fluids, there are proteins in all these fluids, and these proteins will not be able to stay as they were with the change in pH. If the pH goes, the 7.4 pH goes from 7.1 and it goes from 7.6, there will be an absolute calamity inside our body, because with the change, with the lowering of pH, what we see, the more hydrogen ion concentrations are there to damage the proteins, and even if the hydrogen ion concentration lowers, here, there will be a different picture. So our body is strictly maintained in this pH of 7.4, and this reflects the hydrogen ion concentration remains fixed in our body. Our body is a little bit alkaline, our body is a little bit negative or negative on the side. All the proteins are all negative on the side. Alkaline, slightly more alkaline means negative. More negative ions are present. A little bit, the balance is shifted towards more towards the negative ions, and these negative ions, they has to stay that negative to maintain their structure. If you paint them with hydrogen and make them positive, they will not be able to do their functions. The enzymes will not be able to catalyze. The body proteins are structural proteins. Different types of structural proteins are there in our body. Different types of cell contains different types of proteins. All those proteins will get damaged. They will not maintain their structural integrity, and they will happen to lose their structural forms, and they will not function properly. So say pH is very important, and if the pH lowers down, if say I lower the pH, immediately there will be a rise of potassium ions in our blood. Why this type of things happen? So if there is a lowering of pH, say I drink some acid or something like that, the acid goes into our blood, and there is a acidity produced in our body, and this acidity, we don't have to drink acids. We can develop acidity by taking very rich kind of foods, very oily foods, and all those oily stuffs, all those junk foods that we eat. Actually, they lower the pH of our body. They produce metabolic acidosis. They will break down into different types of acids in our body, and they will produce acids, and these acids break down to contribute to these hydrogen ions in our body, and these hydrogen ions will now get into those cells, and these hydrogen ions being positive, the cell has to maintain electroneutrality. You know that every cell has to maintain, if a positive ion goes inside a cell, a positive ion has to come out of the cell. So potassium goes out. Potassium sacrifices his room inside the cell. He goes out of the room. He goes out of the room because hydrogen ions are getting inside the room, inside the cell, and so it displaces the potassium ions of our body, and immediately hyperkalemia develops, means higher potassium levels in the blood. We see higher potassium levels in the blood. So these are the different types of calamities that occur in our body. Our body will not be able, our nerves, we know that our nerves function is very important for our sensorium, for our sensory part. So all those nerves will stop acting because potassium, sodium levels are very important for nerves, and these nerves, if the potassium goes out, these nerves will fail to conduct impulses, because these potassium, sodium levels inside the cell are very important to produce impulses, to produce action potentials, so that these action potentials can be carried by different nerves to our brain, and those action potentials, those neurological damages will help, will not at all help our nervous system, and we will be out of your senses if the pH differs. If the hydrogen ion concentration rises, if the hydrogen ion concentration falls, so pH is a very significant part of our body. So we as humans maintain a pH of 7.4, and this pH is very important, and we will now come to the details of another very important equation, which is called the buffer equation or the Henderson-Hasselbalch equation. And why do we study this buffer equation? This is because there are buffers in our body which helps to maintain the pH of our body. There are different types of buffers. These buffer balances, if there is any perturbation, if there are any changes in the pH, these buffer will now come into play, and they will stop this change. Come on, you have to stop this change. There are these buffers. What they do is that they stop the change in pH. If the pH goes down or the pH goes up from 7.4 to something like 7, 8, 9, and so the pH goes down from 7 to say something like 5, 4, these buffers will now come into play, and these buffers will somehow lower the differences, the pH differences. So buffers counteract when there is a pH change, when there is a sudden metabolic acidosis or sudden metabolic alkalosis, or there is a sudden respiratory acidosis. Why respiratory acidosis? Because if carbon dioxide, what is respiratory acidosis? Say if it's carbon dioxide is increased in our body, and we know that when carbon dioxide increases in our body, we produce more bicarbonic acids. So these acids, like metabolic acidosis, though respiratory acidosis is because of carbon dioxide. So whenever carbon dioxide accumulates in our body, we develop a condition called respiratory acidosis. And these different types of acidosis that develops in our body is counteracted by the buffer systems of our body. And these buffer systems, to understand the buffer systems, we now need to understand what is a buffer. And what is a buffer? Just now I told you, a buffer resists the change in pH. A buffer resists the change in pH. Now what is a buffer? A buffer is primarily a weak acid. Why a weak acid becomes a buffer? That is the main important concept. That is what I am going to teach you now. Say HA is a very weak acid, so it can remain in two forms. It can either ionize, any molecule can ionize, and it can stay non-ionized. It can remain bonded. It bonded to the two atoms of hydrogen, and the anion will remain bonded. But if ionizes, if this anion gets surrounded by water, and water helps this anion loosen its grip with hydrogen, so it ionizes. Say this anion is getting hydrated, and it has to lose this hydrogen, so the hydrogen gets free. This is the ionization part that happens with every molecule whenever it is dissolved in water. The water tends to hydrate, it tends to surround the anion with those positive hydrogens in it. So those hydrogens will now segregate this anion part, and it will leave this hydrogen, and this hydrogen is a very small atom, and this very small atom will now create chaos inside the cell. Now this is because this hydrogen ion cannot be tackled. It will now go on to bind to anywhere. So this is very important. We have to resist this change in pH, and so we are learning buffer now. Well, what is a buffer? A buffer is a weak acid. And what is a weak acid? A weak acid is such a molecule which is always half dissociated, say partially dissociated. It has some unbroken forms, non-ionized forms, and it has some ionized forms. It will always be partially dissociated in ion, and if it is a strong acid will always get broken down into ions completely, and it will liberate more hydrogen ions to create more damage. So strong acids create more damage inside our body, inside everywhere, because it gives more free hydrogen ions, whereas weak acids, they don't do that, and they are always in a half of a 50-50 form. A 50-50 form is that some part of it will always remain intact, and some part of it will ionize into hydrogen and the negative ion. Now, so how does this weak acid perform like a buffer? How does this weak acid perform like a buffer? That is the main thing. That is the principal thing. How do they resist? So if there is an insult to this acid, if there is a hydrogen insult, so if I put on some more hydrogens here, hydrogen ions here, if I put more hydrogen ions inside this solution, what happens here? Because due to common ion effect, some part of it goes, the equilibrium shifts towards the non-dissociated, non-ionized forms. This equilibrium will shift towards left towards the non-ionized form. If I put more OH minus ions here, if I put more say alkali here, say sodium hydroxide, potassium hydroxide, so these are alkalis, they are strong alkalis. If I put some alkalis here, so these alkalis will be now, this acid will see more negative ions, so it will remain, it will now get ionized, more H plus, it will now liberate more H plus ions. To these H plus ions will now bind these hydroxyl ions, and it will get converted to water. So a weak acid remains as a buffer because it can tolerate both these insults. It can tolerate a hydrogen insult, because when there is a hydrogen insult here, when there is a sudden increase of hydrogen ion concentrations here due to some more different acids that I have added, or some different acids that are produced in our body, or we have somehow developed acidity in our body. So if there is an acidity that develops, there is a hydrogen insult, a body buffer will now get undissociated, the equilibrium shifts towards this undissociated form, this non-ionized form, and it will resist the change of pH. It will capture all these hydrogens, and it will come, I'll bind to you, and it will now get towards the equilibrium, will shift towards the undissociated form. And if there is a hydroxyl ion, more negative ions in the solution, the equilibrium will shift towards the right, and all these hydrogen ions that the acid has to offer will now bind to these hydroxyl ions, and it will get converted to neutral water. So this is the mechanism by which a buffer acts. A buffer acts by this mechanism. So now I'll go to a little bit chemistry of these buffers, and I'll have to now refer to you about the buffer. First I'll tell you how this thing operates. Say HA and H2O. HA is an acid. This is an acid solution. This acid is in water, and this HA ionizes to produce H3O plus A minus. Now this can be written as H plus and A minus. Now this is the ionized form of the acid. Now we know that the equilibrium constant of this is KEQ. Now what is this? If I write what is KEQ, we know that it is product by reactant. So it is the H3O plus into HCO plus, into A minus by HA into H2O and this equilibrium constant into the concentration of water, KEQ, into water concentration, it becomes H3O A minus by HA, or it can be written like this: H plus A minus by HA, undissociated form. And this is actually this is called the acid dissociation constant, Ka. This KEQ into H2O is called the acid dissociation constant because it is this acid is now broken down into two ions, and it is called the acid dissociation constant. So what is the acid dissociation constant? It is just so very simple. This HA is now ionized to H plus and OH minus, H plus and A minus, and this is the acid, and this is Ka. And what is Ka? Ka is concentration of H plus, concentration of A minus by HA, product by reactant. Or H plus into A minus, molar concentration of A minus by HA. So this is Ka. If you remember what I have told in your earlier class, what is pH, now I just revise you here. P is the negative logarithm. The p value indicates negative log value of what? Hydrogen ion concentration. What is pH? pH is the negative log value of hydrogen ion concentration present in a solution. What is pKa? pK is just similar to pH. pKa of an acid is just similar to pH in that pK is the negative log. P here also P designates that it is a negative log of the Ka value of the acid. pKa is the negative log of the Ka value of the acid. So more the Ka value, more the acid remains the numerator. If the multiplication of these numerators is if the numerator concentrations are high, so the values of Ka will be high. The numerator is the ionized forms. So if the ionized forms means the hydrogen ion concentration liberated by the acid is more. If it's a stronger acid, the Ka value will go higher. So the Ka value is higher for any acid liberating its H plus ions. The Ka value goes higher. The Ka value is higher. So what is high? Is 1 by 10 to the power minus 10 to the power minus 3 higher than 1 by 10 to the power minus 5? It is just the reverse. 1 by 10 to the power, say 10 to the power minus 3. So what is higher? Higher is, say, 10 to the power minus 3 moles per liter and 10 to the power minus 5 moles per liter. Which one is higher? Obviously 10 to the power minus 3 is higher than 10 to the power minus 5. 10 to the power minus 7 is lower. Why? Because 1 moles per liter means 1 mole is now here 10 to the power minus 3, it's a fraction of 1000, 1 by 1000 moles of hydrogen. So if I write a pKa value which is around 10 to the power minus 3 and I write a pKa value which is around 10 to the power minus 7, so this indicates a much higher pKa value. That means it's a very strong acid. It's a very strong acid and it has liberated all its H plus ions and it has ionized, it has liberated all its H plus ions and it has completely ionized. So if a pKa value is around 10 to the power minus 7, that means that it is 1 by 10 1, 2, 3, 4, 5, 6, 7. So it has given very low amount of hydrogens. It is 1 by 1 mole of hydrogen divided by so many numbers, so many, it is around, say, 1000, then a million, 10 million. 1 by 10, it's just a fraction of 1 by 10 million, 1 by 10 million moles of per liter of hydrogen ions. So it's a much lower concentration of hydrogen ions present here now. So if the Ka value gets lower, that means this 10 to the power minus 7 and the Ka value goes higher, so it is around 10 to the power minus 3 for an acid. So this is a very strong mineral acids like hydrochloric acid, phosphoric acid. These acids have a Ka of around 10 to the power minus 3, while biological acids, say lactic acid, say bicarbonic acid, say carbonic acid, these acids have a pK of around 10 to the power minus 6, 10 to the power minus 7. These are the pKa values. They are much lower pKa values. Now you have understood that what is the pKa of an acid. Now I'll go to an equation which is called Henderson-Hasselbalch's equation. So what is that equation? It is pH is equal to pK plus log of A minus by HA. How do we derive at this equation? I will come later, but this is called Henderson-Hasselbalch's equation. pH is the pKa plus log of A minus by HA. You can say is that pH is equal to pK plus log of A by Ha. A by Ha. Just to remember it. Now, why is this equation so important? Because this equation tells us about the pKa value of every hydrogen liberated every hydrogen has a affinity towards the molecule which it was bound to. Say I have 3 hydrogens bound to H3PO4. I have 1 hydrogen bound to HNO3. I have 2 hydrogen bound to H2SO4. Say I have 2 hydrogen bound to different amino acid. The amino acid have hydrogen. It can bind a hydrogen to its amino terminal. It can bind a hydrogen to its carboxy terminal. So the amino acid, the sulfuric acid, the nitric acid, they will liberate their hydrogens. But with every hydrogen liberation, they will have a pKa value of that hydrogen liberation. The pKa value of a particular hydrogen getting liberated from the molecule is actually what? What is the pKa value? What is the pKa value? The pKa value of that hydrogen is the pH at which that molecule will remain in its dissociated and undissociated form. When the dissociated form and the undissociated form gets equal, say this liberation of hydrogen has taken place, and this liberation of hydrogen has developed into a dissociated form, and this dissociated form of the acid is represented by A minus, the undissociated form represented by HA. So if these two amounts goes equal, we get a value of log 1, which is actually 0, which is 0. Log 1 is equal to 0. So what is the pKa? What is the pKa of that hydrogen, of that hydrogen getting liberated from the acid? It is that pH. At a particular pH, the acid will stay in its 50-50 dissociated is to undissociated form. When the acid stays in its 50-50 dissociated and undissociated form in equal proportions, the dissociated form and the undissociated form stays in equal proportion. Say it's a buffer, it's actually the best buffering part, the best buffering time when the dissociated and the undissociated form remains equal, means it can tolerate a hydrogen insult as well as a hydroxyl insult equally. So then it becomes a very good buffer. So then the dissociated part and the undissociated part goes equal. pK, what is pKa? The dissociated form and the undissociated form gets equal, means the value gets 0. So you don't have to need to worry about that. It is the condition. pK is that pH. So what is the pKa? That particular pH when that acid will have a 50-50 condition of its liberation of that hydrogen. A 50-50 condition. It is a very good buffer at that pKa, and it is in a 50-50 dissociated is to undissociated form. pK is that pH. So by Henderson-Hasselbalch's equation, we can define pK as that pH where the acid is in its best potential buffering form. It is a 50% dissociated or a 50% undissociated state. The 50% dissociated state is actually a 50% undissociated state, 50% non-ionized state. So that pH is the pKa of the acid, of that hydrogen liberated from that acid. So this is a very important implication. This is a very important implication. Why? So if the pKa is a very high for an acid, say it is around 10 to the power minus 3, so it will, so that acid, so if the pKa is around 10 to the power minus 3, at that pH only it is 50-50 dissociated. So if you now it is around pH 3, it is, as I say, if it is 10 to the power minus 3, it is around pH 3. At pH 3 it is 50-50 dissociated. At pH 7, what happens? It is completely dissociated, because its value is, if the pKa value is around 3, so that means 10 to the power minus 3, that means it is 50-50 dissociated at a pH of 3. So that is not the pH of our biological fluid. Our biological fluid remains at 7.4. At 7.4, so it is completely dissociated. So pKa values are very important. So what acids can be a good buffer? Whose pKa values are around the pH values of our biological fluid. Our biological fluids are of all 7.4. Our body is around 7.4. So if a buffer has to be a very good buffer for us, say if you want to serve as a buffer for us, your pKa value has to be around 7.4, then only you can act as a buffer. So at 7.4, you remain at a 50-50 dissociated form, you remain at a 50-50 dissociated and undissociated form, and you can now resist the change of a acid insult or a alkaline insult in our solution. So this is but acid which has a pKa of a high pKa value of 10 to the power minus 3 or at a pH of 3, it will not be of much use to us, because at pH minus 3 we are almost gone, which means all our fluids have got changed and everything, so it will not be of purpose to us. So here, so in this class you have now understood all the details of the pH and the significances of pH, the hydrogen ion concentrations. Now I'll go to the derivation of this equation, what is called Henderson-Hasselbalch's equation. So let's see how do we derive at Henderson-Hasselbalch's equation. So I'll just write the equation. HA dissociates into H plus ions and A minus ions, the anions. So this reaction is in equilibrium. HA can remain in undissociated non-ionized form as well as it can ionize. So if this is an acid and we say that its acid equilibrium constant is Ka, so what is Ka? Ka is the ratio of the products by reactants, and the products are H plus, A minus by HA. So this is Ka. Now if I take log both sides, so if I now equate for H plus ions, if equate for H plus ions, the equation turns into Ka into HA, by A minus So if I take negative log both sides here, so negative log of H plus ions, which is actually pH, equal to negative log of Ka, which is actually pKa, negative log of Ka minus log of HA by A minus. If I just reverse this, if I just reverse this reversal will put positive sign here, A minus by HA. So pH means the negative log of hydrogen ion concentration, the negative log of hydrogen ion concentration that the acid liberates is equal to pKa plus log of A minus by HA. Now What is this implication? This is the Henderson-Hasselbalch equation. This is called the Henderson-Hasselbalch equation. So what it tells, what this equation tells? This equation tells that when the Ka value, this Ka value, the Ka value, the pKa value, the negative log of pKa, negative log of pKa is what? Negative log of pKa, negative log of Ka, sorry, negative log of Ka is pKa, negative log of pKa is log of 1 by Ka, log of 1 by Ka. So what this equation tells is that when the dissociated form and the undissociated form, the dissociated form is represented by A minus and the concentration of the undissociated form is represented by HA, when these two forms equals, so when the acid is now in 50-50, 50 portion of it is in undissociated form and the another 50 portion is in dissociated form, in ionized form. So when this acid is in 50% broken condition, in ionized condition, so this value goes log of A minus is, they can cancel each other, they can cancel each other, and the log of one value is 0. So pKa, the pKa value of that acid, the pH at which the acid goes in 50-50 undissociated form is the pKa. The pH at which the acid goes into the 50-50 dissociated form is the pKa value of that acid. That means the acid is in its highest buffering state. Highest buffering state. pKa indicates its buffering capacity. If an acid has a pKa range of 10 to the power minus 3, it's a very strong acid. Say it will be at pH 3, because it is at 10 to the power minus 3. It is a high hydrogen ion concentration. It can liberate high amount of hydrogen. It is around 1 into 10 to the power minus 3 compared to something which has say 1 into 10 to the power minus 7. So 1 into 10 to the power minus 7 is a much lower hydrogen ion concentration than 1 into 10 to the power minus 3. So acid at a pKa of say 10 to the power minus 3 will have its buffering power at pH 3. Acid at 10 to the power minus 7 will have a buffering power at pH 7. So those acids which have their pKa values at around our biological fluid pH, our body pH, our body pH is slightly alkaline 7.4. So they will be a very useful acids, and those acids are very useful for us. And why they are useful? Because they can act as very good buffers. They can act as very good buffers, and they will resist all the types of acidosis that develops in our body. They will resist all the type of alkalosis that have developed in our body. So a kind of acid insult, alkali insult, they will resist that, and so they can act as a good buffer. So so the pKa value of an acid, of a biological acid, say lactic acid, say bicarbonic acid, say any other acid of our metabolism, say all these acids that are produced in our metabolism, in the glycolytic cycles, in our Krebs cycle, all those acids, phosphoglyceric acid, diphosphoglyceric acid, pyruvic acid, all these acids can act as a buffer if they have a pKa of around 7.4. A pKa value signifies its half dissociated form. The pH at which it remains in its half dissociated form is the pKa value of that acid. Thank you for listening to me.